Information is not matter or energy, but often it rides upon matter and energy.
Explain that.
Take a paper book. It's written in ink molecules on paper, but the information in the book did not come from the ink molecules. It required the author to have the ink molecule arranged into letters and words and paragraphs, but it did not come from the ink.
We've all read that billions of years ago, there was a primordial soup.
There's no geological evidence that such a soup ever existed on planet Earth. They're relying on a scenario that has no geological evidence whatsoever.
We've all read in science textbooks that billions of years ago, there was a primordial soup of chemicals that over those billions of years combined and recombined, and from these random chemical reactions, the first cells emerged, and we call this abiogenesis, life coming from non-life. Now, Dr. Sarfati, I know that you have a PhD in chemistry from an accredited secular university. You've written some of the best-selling creation books of all time. So, to start off, talk to us about chemical evolution. Is this the same as abiogenesis and the origin of life?
Well, first of all, I want to say that the best-selling creation book is the Bible. Okay. Now, yes, I am a PhD chemist, and in fact, some of what I've done in the lab actually is very relevant to what we're talking about. Now, abiogenesis is also called chemical evolution. In fact, that's an official title in many scientific papers, even academic departments with the title chemical evolution. And it's the idea that life came from non-living chemicals. But in my experience, the chemistry goes in the wrong direction in many different respects.
Okay, and we'll get into that a little bit further on. But also some people will say that the issue of life's origin is separate from evolution, and evolution doesn't need to account for it. What do you say to that?
Well, I mean, that would be news to many of the biology textbooks around the evolutionary biology textbooks, which often have a discussion of the origin of first life. You read Richard Dawkins' famous Atheists and Evolutionists. He would always discuss origin of the first life in his evolutionary propaganda books, okay? And the general theory of evolution as defined by G.A. Kirkut from Southampton University, who's an evolutionary biologist. Well, he's dead now, but he defined the general theory of evolution as the theory that all living things evolved from a single-cell creature which itself came from an inorganic form, i.e. non-living chemicals. And also to have evolution, Darwinian type evolution starting with natural selection. Natural selection is defined as differential reproduction, which really means this thing is fitter than this thing, therefore this thing will leave more offspring. So what you need to have something, these two things that will reproduce. So you must explain where that those reproducing things came from. Otherwise, Darwinian evolution is dead on the starting block and materialism is dead in the water.
Right, that makes sense. Well, one of the icons of evolution in some ways that people will point to talk about the origins of life is the Miller-Urey experiment. Mm-hmm. Can you talk to us about what that experiment was and whether it can in fact explain the origins of life?
Well, the Miller experiment was published in 1953, quite an interesting year. That's when the DNA double helix structure was published, and also when they climbed Mount Everest and when Queen Elizabeth II was coronated. Okay, now what they would do, they sparked. It had a mixture of gases, and they applied either UV light or electric discharge, and then they collected the results in a trap, and using very sophisticated chemical techniques which had been invented, they could find traces of amino acids, which are the building blocks of proteins. But I think the Miller-Urey experiment is good evidence of how life could not have happened.
Okay.
There are several things wrong with it. For instance, they use what they call a reducing atmosphere, which means rich in hydrogen-containing chemicals like methane, ammonia, hydrogen itself, okay, which was on Jupiter and Saturn, but there's actually no evidence that it was ever there on Earth, okay? Now they're pointing towards carbon dioxide and nitrogen, which are much more inert and much less favorable. And also there seems to be evidence that oxygen was there just almost from the beginning. Even if you look at evolutionary dating methods of very old rocks, there's evidence of free oxygen. Now, oxygen is a very reactive chemical. We don't think about it. We need oxygen for life, but in fact, we have machines, mechanisms to deal with excess oxygen. Otherwise, it would poison us. I mean, antioxidants, you're familiar with those, okay? And oxygen in that mixture would stop anything from forming and would destroy anything that had been formed. But one paradox is that you need oxygen to have an ozone layer. Okay, so if you didn't have oxygen, you have the ultraviolet light pounding everything to smithereens. Okay, that's one issue. And the other thing is they had to have a trap because the very energy sources that produce these things would destroy them even quicker. And is that really what would occur on the primordial Earth? See, even the ocean would not be much of a trap because ultraviolet can penetrate to tens of meters deep. I mean, you can get sunburned while swimming. You can get sunburned on a cloudy day. So water is not very good at protection from UV. Okay, and that's incidentally an argument against the canopy theory being so protective. Well, it actually wouldn't be. It's not a very protective shield against ultraviolet radiation, which would destroy those chemicals. And especially if you argue for millions of years, because millions of years means more time for destruction to occur.
Right. So what I'm hearing you say is that some of these conditions that were part of this experiment to create the building blocks of life, those very conditions, if they were not carefully managed by, somebody in a lab, for example, those very conditions would actually destroy everything that they were trying to create. That's
a real problem for them. And another thing is no one will take the results of the Miller experiment and try to do the next step because even they found traces of amino acids, they were a grossly contaminated gunk, extremely dilute, and the contamination would stop reactions going on any further. Okay, as I say, they needed sophisticated chemistry to even separate the amino acids so they could detect them, okay? But you haven't got that on a primordial Earth. You've just got the amino acids in all sorts of gunk, which would stop life, stop the process even leading to life.
Right. So basically, there's so many other byproducts that could potentially react with one another in ways that aren't going to form the amino acids that are necessary and everything else.
Right. I mean, you want amino acids to react with each other to form proteins, but they're going to react with all the other things in there, and that's going to stop proteins being formed. It's going to take them out and therefore make them unavailable for protein formation.
Okay, so we've got to ask, could this experiment produce life?
Well, no, they never use that because what they will do, okay, they say, we're here, the Miller experiment produces a trace of this amino acid, so let's buy this amino acid from a chemical company and then do the next step in the reaction and produce a trace of another chemical, maybe two or three amino acids lined up together. Oh, let's go and buy that from the next chemical company. So at every step of the way, they are buying the stuff, and therefore they're admitting the steps it would take to concentrate and purify the material in time for the next reaction. A primordial soup could not do what these chemical factories do that make these pure chemicals.
Right. Yeah. So they're taking, correct me if I'm getting this wrong, but they're taking a look at what's in this primordial soup. They're looking and saying, oh, there's little trace elements of each of these chemicals that are necessary for life. Mm-hmm. Well, let's just go out and buy, a truckload of that stuff and mix it together without accounting for the fact that that's not how this would have happened in real life.
Yeah, you'd need to actually take the results of the Miller experiment and see, well, let's do something with that, and then you might have a case.
Okay. So part of the narrative, at least the way it currently stands, is that this happened in the ocean.
Yes.
So could chemical evolution have occurred in water?
Well, in fact, water is the last place I'd want to try to build up the big molecules from the little ones because, for instance, when two of the amino acids form, it's called a peptide bond, and the protein is just a continuation of that. See, every time they form that bond, they kick out a water molecule. So the process is called condensation, and it's polymerization because they're making many different things as a polymer. Okay, so condensation reaction. But the thing is, you don't want water there, because if there's water around, the water would pop back and actually break up that bond. So it would go in reverse. It's called hydrolysis, which means breaking up stuff using water. So hydrolysis would be what would happen if you have water in the reaction. And the thing is, when you try to make a protein or a nucleic acid, a synthetic chemist will not do it in water. And in fact, what he would often do is add a water-removing chemical. They call them condensing agents to pull the water off, you see. But the primordial soup didn't have those. And even a condensing agent would not last well in the ocean, because the whole point is to react with water, so it's not going to last very long in the ocean. So, in fact, a little anecdote here, giving some creation talk. I think it might have been university, and a guy introduced himself to me in the break about how he's actually a protein chemist, a synthetic chemist who makes these big things. And so during the question time, I actually asked him personally, I mean, would you ever use water in your reaction mixture? And he sort of, of course, in those sort of things, I don't ask a question unless I know the answer. Okay, and he sort of said, "Are you kidding me?" Mm-hmm. Well, that's the whole point. Yes, that's the whole point. I was kidding him. That water is a ridiculous place to do any sort of condensation reactions.
Got it. And I know we're about to start getting into some of the more technical stuff, so we might have to define some of our terms. Mm-hmm. Can you talk a little bit about polymerization and why that's important?
Well, it's a polymer. Polus in Greek means many, okay? So mono means one, so monomer. And you combine lots of monomers and you get a polymer, okay?
Okay.
And the monomer for a protein is an amino acid. The polymer is the protein itself. So DNA, the monomer is called a nucleotide, and they're made up of simpler things as well, three different components. And the polymer is either RNA or DNA. So polymerization is what you need to produce the big molecules of life, the proteins, nucleic acid, but also polymers of sugar, for instance, so like starch and cellulose are polymers of sugar, okay? Mm-hmm. And you have lipids, which are basically lots of hydrocarbon things, okay? That's a fatty stuff. Fats and oils are called lipids, okay? But the main things we want to talk about are proteins, which are the machines of life, and the nucleic acids like RNA and DNA, which are involved in the information storage and processing systems of life. And both of those are polymers, and therefore you need to explain how polymers could have arisen from the monomers they assume arose in a Miller-type experiment.
Right. So unifunctional and bifunctional molecules are really important to this discussion.
Okay.
So can you talk to us a little bit about those?
Okay. I talked about the monomers, the individual building blocks. Like amino acids, they have a functional group on both sides, okay? Mm-hmm. Technically, it's an amino acid one side, carboxyl on the other, and they bond with an amino acid. So two amino acids like this, and then they bond on this one and on this one, okay? So you need to have something which is bifunctional, has two functional groups, okay? Now, if you have something with one functional group like this, okay, the protein bonds, but it can't grow on this end. So this would be a chain terminator, any monofunctional group. And I've actually had a communication with a guy who makes polymers, and he affirmed that even 0.5% of the unifunctional would block the chains off and make the reaction impossible. And this is something I've done myself, because there are various ways you can make nylon in a chemistry demonstration sort of thing. You have two different bifunctional things combining, but you put one in oily stuff, one in water stuff, and where they meet, nylon forms, and you can take off that film and make it into a fiber, and more as the film is drawn off, more comes, okay? And I showed how you could sabotage that by putting a drop of unifunctional there, and the film broke, and you couldn't draw any more nylon out of it. Okay, so I've done this myself. But the problem is the Miller-Urey experiment produced about 5 times more of the unifunctional than the bifunctional. When I said there's about 0.5% is enough to stop the polymers growing because it blocks off one chain, imagine what 5 times as much of the things would do. You would never get very far at all before it came and blocked off the chain. I mean, if you look at the results of what they produce in the Miller experiment, the biggest thing by far is formic acid. The Latin word for ant is formica, so formic acid is found in ant stings. So if you were reporting that experiment, you could say, well, here we found a way of making formic acid. That's what it would be if you were objective and didn't have an agenda. But the formic acid, it's a chain terminator, so it would block the proteins forming.
Right. So what you're saying is that basically for polymerization to occur so that we can convert these monomers into polymers, it needs to happen in those two directions.
Yeah.
But if you have any amount of a substance, really a very small amount, like 0.5% you were saying, or like you put a drop in, if you drop any amount of that substance in there, it's going to stop that reaction and the whole thing is literally dead in the water.
It is, correct.
So on that subject, let's talk about cross reactions and why they're important to this discussion.
Well, see, any chemist trying to do a multi-step synthesis, trying to make something that requires a lot of different steps, at every stage in the way he will try and purify the next step, because otherwise it will react with the other chemicals in the mixture, which you don't want, because that would mean that it takes it away and therefore can't do the reaction you want, because it's doing the reactions you don't want. But the Miller experiment produces a gunk of stuff that would cross-react with the things that you're wanting to form proteins that react with other things, and therefore, again, would not produce life. And that's why, at every step of the way, these chemical evolutionists trying to show how life could have arisen without intelligence, they will actually buy the stuff from a chemical factory at every step of this proposed sequence.
Right. So to actually get the things to react that we need to react, it takes quite a bit of human interference and purifying of the actual chemicals to make that happen.
That's correct.
So we have to ask, why can't sugars and amino acids form in the same space?
Well, they have mutually incompatible conditions to produce them, and I'll go into that when we talk about how to produce sugars. But also, they actually undergo a cross-reaction, because the amino acids have an amine group on one side. The sugars have what's called a carbonyl group, and they are well known for reacting with each other to form what's called imine group, okay? But that is what causes browning in foods. They're called Maillard reactions. Okay, like when you bruise an apple, what happens? You're breaking some cell structures, and therefore some amino acids and protein and sugars can now come into contact and produce that browning reaction. And when you toast bread, that's the sort of thing that causes the browning of bread when you toast it, okay? But the problem is, in a primordial soup, you don't want that to happen. You want the sugars to be free to form, say, the DNA or RNA. You want the amino acids to be free to form proteins, but if they're reacting with each other, they're not free to do anything more in the direction you want. So sugars and amino acids could not come together. They'd have to do their things, form the amino acid, the proteins on one side, nucleic acid on the other side, and then come together after that somehow. But before then, they'd have to be in separate parts of the primordial soup, basically. Otherwise, they would cross-react, and that would be fatal.
Right, and the whole problem with the primordial soup in this case is that it's a soup. It's all just kind of a slurry blending together. And
by the way, just as an incidental thing, there's no geological evidence that such a soup ever existed on planet Earth. I mean, you'd expect to find certain traces of such a thing. There's certain chemicals you'd expect to find if such a soup had ever existed, but there's no trace of that in the geological record. So they're relying on a scenario that has no geological evidence whatsoever.
Right. So how far have they got with forming life in a lab?
See, when they try and form life in a lab, they actually skip a lot of this chemistry stuff, and they start very advanced. I mean, Craig Venter's group has made a sort of synthetic life form called Cynthia. Now, it's a very vulnerable thing because it's such a simple life form. I mean, it wouldn't last very long. Okay, but what they had to do is they take the genome, the DNA sequence of a mycoplasma, the simplest self-reproducing thing we know. And by the way, that's a parasite. It's so simple, it needs more complex things to live on to get the stuff it needs. And one theory is, in fact, the mycoplasma is a degenerated form of something more complicated. It's lost a lot of its genome. So could that have been the first life? It doesn't seem to be. Okay, but they use that, and they have the structure of an actual cell, and they even use enzymes also from previously living things. So did they really create life from scratch? But let's grant that they did. Imagine they could go further in the future and start the genome, the cell structure, the enzymes, and make them all from scratch. But how would that show that life could have arisen without intelligence? Because an enormous amount of intelligence was needed to make that self-reproducing cell, which seems to show that you need intelligence to make life and not bundling chemicals. And I'm talking about bundling very advanced kind of chemicals, not just the simple stuff that you get in the supposed primordial soup.
Right, yeah. It would show that you have to carefully husband all of your resources and time everything and stop those reactions just to get even a very, very simple form of life to function on its own.
Right. They started very advanced. Let's make it very clear. They started extremely advanced and very far on the step towards life, almost there, basically.
Yeah.
Not down here at the primordial soup level.
Right. So what they did is almost more like genetically engineering rather than creating a new form of life.
That's basically what it was, yeah.
How far have they got at forming sugars in a lab?
Well, okay, you're familiar with sugars as you stir it in your tea or coffee, maybe. Okay, but sugar is actually a fairly big class of chemicals. They have some similarities. Now, the thing is, DNA has a sugar called deoxyribose. RNA has a sugar called ribose, okay? And also glucose is important for metabolism. So sugars are important for metabolism and the information storage. So you need them, but in fact, sugars are quite unstable. And we're talking about sort of years rather than millions of years, sugars I'd expect to break down in a primordial soup. Now, how do they make them in these supposed simulations? Well, there's a reaction called the Butlerov or the Formose reaction. And what they do is they take formaldehyde, which is a simple thing. It has one carbon atom. They have alkali or base, and that makes the formaldehyde join together and form sugars. Now, one problem is that it forms a confusing jumble of different sugar types, okay? So to get RNA from that, you'd have to somehow separate the ribose from that confusing jumble and do something with it, okay? But the other thing is that alkali helps to break down sugars. You see, you needed alkali to produce sugars, but alkali also breaks down sugars. There's a reaction called the Cannizzaro reaction, okay? And therefore, to make sugars, you'd have to stop the reaction at the right time before the Cannizzaro reaction destroyed the sugars that you made. So the very same conditions that produced them tend to destroy them as well.
Right. So what I'm hearing you saying is that sugars are foundational to every aspect of life. They get used in so many different types of molecules, but the conditions that would create them if left unattended will basically destroy them.
Correct, yeah. And yet one of the most popular theories of origin of life is called the RNA world, which they assume that an RNA molecule could do both the machinery of life, like enzymes do, as well as the information part of life. But to get RNA, you have to have sugars. And that itself is enough of a problem for the RNA world. So it's interesting, a biochemist in New Zealand even wrote a paper called Why the RNA World is the Worst Theory of Origin of Life, except for all the others.
Right, and you almost run into a chicken or the egg problem with it sounds like.
Oh, very much so. A lot of those around, actually, yes, when it comes to origin of first life.
Talk to us about chirality and why it's an issue.
Okay, well, see, chirality comes from the Greek word for hand, okay, and it refers to things like your left hand and right hand are mirror images of each other. They're not, you can't superimpose them, okay? Only a right-hand glove will fit on my right hand, okay? Because they have different spatial orientation. So you can't mix the two. Now, the primordial soup, a Miller-type experiment is going to produce a 50-50 mixture of right and left. That's called a racemic mixture or a racemate. That's a technical term, okay, but it means a 50-50 mixture, okay. But for living things use exclusively one hand, the same hand, hence it's called homochiral. Like proteins are made of left-handed amino acids. The DNA, RNA, your metabolism uses exclusively right-handed sugars, okay? You need to have exclusive one-handedness for the molecules of life. You see, the proteins, the enzymes, are often tuned to atomic level precision, and you couldn't get that if you got the other hand throwing off the orientation. And when it comes to DNA or RNA, they have to form a double helix, and they also have to bond with the other. So it's a double strand. They have to have a sort of what they call base pairing. And that again needs to have this spatial orientation right. And what they've done is try to make an RNA on an existing RNA template, and they found they just had one of the wrong-handed molecule. It threw off the orientation, and the chain just would not keep growing anymore. So it's a chain terminator. Just as one of these wrong-handed things would be a chain terminator. So when you might see things like, we've produced what they call a chiral excess of a tiny amount, that's not enough, because you need basically almost 100% for the proteins and the RNA to work, not just a little bit of one or the other. And yet another problem with this is that over time, this homochiral, one-handed mixture will revert to the racemic mixture of 50-50. There's even a dating method. It's called amino acid racemization dating, which supposedly measures age by how much this process has occurred.
So what I'm hearing you say, and you can correct me if I'm wrong, is that basically we have these molecules that are physically shaped in two different ways, and there's basically one, we can call it left-handed or right-handed in my case, and one that's left-handed. And basically, in order for life to form, you need essentially all of them to be of one hand or the other, because if you drop even a small amount of the other-handed molecule in there, it's going to throw off everything and stop the reaction, right?
Well, in the case of forming, say, RNA on a template, because it will throw off the helical structure, it means it won't pair up properly. It can no longer pair up, and therefore the chain terminates. It can't grow anymore in that direction. That's a very serious problem. Just a single impurity of the wrong hand would do that.
Right.
When it comes to proteins, you've got a little more leeway, but not very much, and that's why almost all proteins of living things are left-handed, because again, the protein machines, the enzymes, have to be tuned to atomic level precision. Okay. Another thing is that alkali is very good for making proteins racemize. Okay,
and you have to refresh my memory. What is an alkali?
Well, you can think of an acid. If you want the technical stuff, an acid and alkali are opposite each other.
Okay.
Acid is something that donates a hydrogen ion, it donates a proton. An alkali is something that picks it up. So they're opposite, okay? Now, alkali, remember I said that alkali was needed to make sugars. Okay, it also helps to destroy sugars once they're made, but also alkali helps to undo the handedness you build up.
Okay.
So again, another example of incompatible reaction conditions to make the sugars and to make the proteins, because the alkali is just what you don't want when you try to make a protein because of the racemization, going to 50-50 mixture when alkali is present.
Right.
President.
And so to clarify, when we're talking about racemization, what we mean is we have our one-handed mixture, let's say it's all right-handed molecules.
Yep.
Over time, it gradually actually becomes 50-50.
Correct.
And this is a problem because that represents a contaminated mixture and you're not going to get the reactions that you need.
That's definitely correct there, yes. And I have a personal experience in a chemistry lab of this thing because I made a certain inorganic compound that has two mirror image forms. And of course, when I made that, it produced a 50-50 mixture. That's everyone knows that. Mm-hmm. And so how do you separate them? Well, what you have to do, think about a pile of gloves. You couldn't separate left and right by hand with a pitchfork because as far as a pitchfork is concerned, they're all the same. Mm-hmm. But if you put my right hand in, it would hopefully fit only the right-handed gloves, okay? And that's what you have to do in a chemistry lab. You have the 50-50 mixture, but you introduce a one-handed thing, homochiral stuff, and that reacts preferentially with one hand or the other. And where do those things come from? They come from living things. Okay, so it's interesting. Okay. Where did the first chiral stuff come from? Because when we do a chemistry lab, we're actually assuming the chirality is already there from living things, okay? So that's how I separated the two. But then you add activated charcoal to the mixture, and it quickly went back to the racemate, the 50-50 form. And this is what would have happened even if you somehow got a homochiral mixture, a one-handed mixture. Over time, it would go to the 50-50 form, as well as if you had some catalyst like charcoal or a catalyst like alkali, as I mentioned, it would make that happen quicker, and high temperature would make it happen quicker as well.
Right. So we're saying that even in a lab, in order to get that homochiral mixture, we have to do some chemistry that's pretty precise and actually deliberately try to do it. And the tendency over time anyway is going to be to return back to that heterochiral mixture.
Well, that's right, which is also called a racemic mixture. And you see, one theory of origin of life is alkaline hydrothermal vents, which means you've got extremely superheated water coming through the ocean.
I've heard this one.
But as I say, high temperature and alkali are just what's going to make it racemize. So again, that's the wrong sort of environment to try to build up a homochiral protein. It's just totally the opposite of what you need.
Exactly. Okay. Well, let's shift gears a little bit and talk about DNA, because we've been talking a lot about the basic building blocks and the conditions that need to happen to turn those into something complex like DNA. Why can't evolution explain coded information within DNA?
Well, first of all, let's talk about DNA. That is the coding molecule of life, okay? It's quite an interesting molecule. It stands for deoxyribonucleic acid. Now, remember what I mentioned, acid is something that gives off a proton or hydrogen ion. It gives off something positively charged. Therefore, what's left behind is called a conjugate base. It's negatively charged, and that's a force field that actually helps to protect it against certain attackers that would otherwise destroy it. It repels those things. So it's actually a good design feature that our information is coded in an acid, okay?
And so it protects it from damage, basically.
It is very good protector, but even with that, DNA is a very unstable molecule. I think people don't realize that, but if you check out the Nobel Prize of 2015, and the Nobel Prize site itself says that you have Thomas Lindahl, who's a chemist. He realized that DNA breaks down really quickly.
Okay,
you could measure that. That's in the lab. And therefore, how could life survive if DNA is so unstable? He realized there must be incredible repair machines to undo the damage, because every cell in your body probably has 3 million letters worth of damage every day. Out of the 3 billion letters you got a million every day. So you have to have repair machines to make sure that's not going to happen. But of course, a primordial soup wouldn't have repair machines.
Right.
No, that's a problem. And the thing is, DNA has the instructions to build its own repair machines. Yeah. But unless you had repair machines, those instructions would be degraded. And therefore would not make the right repair machine. So that's one of the chicken and egg problems, isn't it? Which came first, the DNA or its repair machines, because they all have to function together. Okay, so DNA, as you rightly said, is the information storage of life, but as I say, it's unstable. Now, there's another molecule called RNA, which is about 100 times less stable.
Okay. So it breaks down even faster.
Yeah. So the idea of an RNA world, our first life being an RNA, that's chemically preposterous because of how unstable RNA is. Okay, but yes, it is an information storage system. Okay, and it actually has the instructions that make us who we are, has the instructions for the machines of life, okay? And also it's in a way that these instructions can be copied to the next generation. Say you didn't really get your mother's eyes or your father's ears. In fact, it was the instructions of your mother's eyes and father's ears that got copied to your DNA when you began life as a fertilized cell, okay? So it's a very important molecule for life. But the thing is, where does the information come from?
Right.
See, information is not matter or energy, but often it rides upon matter and energy, but is not from the matter and energy.
Explain that, yeah.
Okay. Well, see, your computer screen, or take a paper book. Okay, take a paper book, okay? It's written in ink molecules on paper, but the information in the book did not come from the ink molecules. I mean, you can't go splat with ink molecules with pouring ink on a book and get information out of it. It required the author to have the ink molecule arranged into letters and words and sequences and paragraphs, okay? But it did not come from the ink, okay?
Okay.
In the same way, there's nothing in the chemistry of the DNA letters to make them write the messages of life. Okay, so again, just as information in the book comes from outside the ink, so does the information of your DNA come from outside the chemistry of the DNA. Okay, it's not caused by the chemistry, it's something external to the chemistry. In fact, the chemistry has to be free to do what it needs to do, otherwise you wouldn't have information. You need to have the flexibility for information to work. If it's deterministic, if it has to line up a certain way, you could not use that as an information storage. If ink only formed a certain way on paper, you could not use it to write a book. You'd have to have the flexibility.
You could write one book, and that was it.
That's right. And the other thing, my books aren't any use to you unless you know language, right? You have to know English. And again, DNA, you had to have certain languages to make sense of the instructions.
Right. So the DNA, not only does it contain, we've talked about how it contains the instructions for life. We've talked about how DNA contains a lot of information, and how that information not only contains its own repair machines and instructions on how to build those, but also contains instructions for translation.
Well, yeah, because see, if DNA stores the information for reproduction, but it needs a lot of machines to reproduce. DNA doesn't copy itself. It needs an incredible array of protein machines to do that, and DNA has the instructions for its own reproducing machinery. But once again, if those machines weren't working from the start or were working less perfectly, then the reproduction would not go as well, and therefore the next set of instructions for those reproducing machines wouldn't work as well, so make even more errors. That's called an error catastrophe. And things would grind to a halt really quickly unless machines were basically almost perfect right from the start. And this is what God tells us in the beginning. He created things very good. And scientifically, we realize they have to start basically very good, perfect. And what we see is it's deteriorating. It's coming downhill. It's not coming from down here because it couldn't be down here because it would actually get worse. It would never get better, but actually everything would degrade if it was down here. Another thing is that DNA, well, think about it, it's several feet long. And in your cell, you've got a microscopic cell, and your DNA might be 2 nanometers in diameter. The thickness of it, very thin, but it's feet long if it was straightened out. So you think of a coil that's that long, and think of the old-fashioned, telephones. I mean, Gen X's and Z's wouldn't know that, but yeah, they get tangled up. So in fact, you have these detangling machines to take care of that.
Basically, they're going through and straightening it out so that...
Well, it's more than that. They're called topoisomerase. They always have to have these fancy names. I don't know why. Okay, but what it does, it snips it actually. It snips the DNA and then sort of rearranges it and then puts it back together. It's it has to have all those steps working perfectly. Otherwise, it would be useless. So evolution would have to explain how all these 3 steps would happen at once, because 1 or 2 would actually make things worse. Okay, but the thing is, DNA has the instructions to build those detangling machines, but you'd need the detangling machines to read the instructions to build the detangling machines.
Right. It's
to
a
reproduce
circle.
them. Well, for instance, there's an antibiotic class. It's called fluoroquinolone. Okay, that's a type of antibiotic that you have, and what that does is stops these machines from putting the DNA back together again.
Oh, okay.
Well, that's a problem because it goes through, snips the DNA apart, but then doesn't put it back together again.
And that's the end of it.
That's the end of it.
And that kills the organism.
Yeah, so that's how antibiotics often work by targeting these machines in living things, and just has to target one type of machine, the cell's gone.
Right.
So which shows you how so many of these machines must be needed for living things to work. If we can just remove one of those machines and it's enough to kill the cell. Okay, so this is one example. One of my favorite machines of living things is those topoisomerases, because of how complex and how vital they are for living things.
And this is, when we're talking about, antibiotics targeting these machines. This is an example of irreducible complexity at work, right?
You could say that, which means that's a phrase invented by Michael Behe of the intelligent design movement. We interviewed him in Creation Magazine maybe before you were born, I don't know.
Yeah.
Okay. And he invented that term. Another term, which is sort of more functional, Dr. John Sanford, who's also a friend of our ministry, he refers to a functionality threshold, while the irreducible complexity emphasizes the actual structure, while Sanford's term emphasizes the function. But they sort of mean the same thing. You have to have a certain function before it can even be working in the first place.
Can you break that down a little bit more for us?
Well, okay, so the topoisomerases has to do three different things, and if it didn't do all three, it would actually do more harm than good. So until it got to that level, it may as well not be there.
Right.
Okay. And by the way, in fact, even talking about natural selection building up these machines is not possible, because these machines are necessary before reproduction can occur.
And so we need them for natural selection to even be taken into account.
Yes, you can't use natural selection to explain anything needed for reproduction. And topoisomerases certainly are needed.
Right. So with DNA as a whole, we've talked about it as an information system several times throughout this conversation. But is that really what it is?
Well, it definitely is what it is, because even talking about languages, in fact, your book is only in one language, but DNA has multiple languages on the same set. So that's where, the RNA, the related chemical, is much more unstable, but it's actually dynamic enough to be a control system. And it's the RNA that actually controls how the DNA is decoded. Okay, so the language most people are familiar with is a language from DNA to proteins. That's one language. But you also got language on how the proteins, which ones are made and which are not made, and also things about where it is in this spatially located in the cell, and even as far as the time is concerned. Okay, I mean, think about your eye cell. Okay, your eye cells have bone and muscle information, but it's all switched off.
Interesting.
Yeah, but which means the eye cell probably have the information for making eyes so it's spatially close together and accessible to the machines, and the bone information is turned off but also buried because you don't need it, you see. So
but it's still there.
It's still there, you see. I mean, all your cells have all the information, but a lot of it's turned off. When that doesn't happen, that's called cancer. Okay, it's not good for you. Okay, so cancer is when the cells multiply uncontrollably, and that's what single cell things do. They just multiply. Multi-cell things, they have to have some control over that, so it doesn't. So they have to work together and don't crowd each other. Okay, that's one issue. And the so-called junk DNA that people talk about, often that's involved with these other languages.
So it's not junk.
Well, it's not junk. I think a lot of people are now saying junk DNA is a science stopper, you might say. It stops science because if you think something's junk, why bother researching it? And so for decades, people didn't work out the incredible function that the junk DNA has, because the ENCODE project showed that about at least 90% of DNA is transcribed into RNA. And then the words RNA copy of the DNA information is made, which means it's doing something, which means it's part of the operating system. And the Australian geneticist John Mattick, he said, last millennium that the junk DNA is the biggest blunder of molecular biology.
Right.
And then he became the head of the Genomics Institute in the UK, and he thought that was his most important contribution, is realizing that the so-called junk DNA had incredible functions as part of the cell's operating system. It's really interesting. I mean, think of a book, language shows you've got intelligence behind it, at least human level intelligence. But what about multiple languages in the same book? That would seem to demonstrate a superhuman level of intelligence.
Right.
A book you could read in English and German and read backwards and read every 10th letter, and you get a coherent message for every one of those languages. That's what you got with DNA. Multiple languages on the same part of the DNA.
That's incredible. So I understand that at one point you engaged in some debates with a head organic chemistry professor on this.
Yeah, this goes back to my university days when I was doing my PhD, maybe before you were born, okay? Now the head of organic chemistry, who is an expert, a world-class expert in carbohydrates, in other words, sugars and the things coming from it, he gave 3 lectures to a introductory class on origin of first life, okay? And he invited me to have 1 lecture, just 1 to his 3, to try to take it apart. But he admitted afterwards that if a vote was taken, he would surely have come off worse. He realized I'd made some major hurdles to his theories. But in fact, it wasn't that hard because I used the chemistry I'd learned from him.
Okay.
In his courses, he taught me a lot of the chemistry I used. So I was using the real science he had taught me to show why his chemical evolutionary scenario didn't work. It doesn't work against real chemistry. The real chemistry I learned from him shows that his basic blind faith position of life from non-living chemicals just couldn't work.
So it sounds like you were using the very same logic and science that he himself had taught you to demonstrate that the biblical position was more plausible than his.
That's correct, yes.
Well, Dr. Sarfati, this has been great, giving us a huge amount to think about and dig deeper on. Where can Christians go to learn more about this topic?
Well, for this topic and other scientific refutations of evolution, I can't recommend anything more highly than the video called Evolution's Achilles Heels. It's a bit over 90 minutes long, but it's in 9 separate sections, and it comes with a free study guide. And to discover, it talks about lots of different aspects, including origin of first life, which I do a bit of talking in that one. But it also even ends with the ethical and moral implications of your worldview, goes into the evolutionary foundations of the Holocaust, for instance. It's interesting, a long-age Christian apologist, Frank Turek, actually played a clip from Evolution's Achilles Heels when he debated an atheist about the need for a God to have foundation for morality, but he describes me as a Jewish man talking about the Holocaust. Well, I am Jewish, actually. I'm also a Christian, a Jew who believes in Jesus. But interesting how he used this very much short age, young Earth creation video in his talk, okay, in his debate with an atheist, and cites me in that one, okay. But it also has, they say, geology, genetics, natural selection, radiometric dating, and even cosmological evolution is the Big Bang, right? So it has a huge amount of material, and it comes with a free study guide.
And for sticking with us, we are going to make that 90-minute award-winning documentary available for a free 7-day stream in the description below. Dr. Sarfati, thank you so much.
Well, thank you, Sheridan.